everyone. Welcome to another episode, or the next episode, where we left off in our year 2 biology discussion, right? Where we left us off was this discussion about, we started talking about mutations. And what we were talking about there was the idea that changes to the DNA would result in changes to the mRNA. Remember, we're referring here to the sequence of bases, right? And we said previously we said that any changes to the sequence of bases in the DNA, i.e. mutation, would result in a change in the sequence of bases in mRNA, and potentially this could change the sequence of codons, and potentially this could change the primary structure of a protein. And if the primary structure of the protein changed, then potentially this could change the tertiary structure of the protein, changing its shape and changing its function. And like any random change to a highly intricate machine, any random change, usually, like 999 times out of 1,000, 999 times out of 1,000, it will have a negative impact on the function. There is the potential of some positive change, but this is what I wanted to talk about next, was considering all that story, what are the types of changes that we could have to the DNA, and what is the range of the impact it can have on a protein's function. Okay, so this is the topic of our discussion. Yeah, I'm going to keep it short. It's just a little opportunity for us to revise. Okay, remember, the tone of this is I'm going to keep it quite simple. Get comfortable, maybe get something to drink, just listen and hope it makes sense to you. And if it doesn't make sense, all the better, because that means we've identified something that you might need to work on then, which otherwise we might not have identified. Okay, so let's do this. We're talking about the mutations, right? So what we discussed last time was the impact of swapping a base out. So let's just quickly talk about the types of mutations. What are the types of mutations that we could have? And there are mutations that are bigger, chromosomal mutations, for example, that affect entire chunks of chromosomes. I'm not really going to talk about that right now. I'm talking about changes that could happen within one particular gene. Okay, so we could have a substitution. We could have insertions. So by mistake, a base just gets added, so an insertion mutation, or by the same topic, or by the same concept, we could also imagine that we could get a deletion. Okay, so these are the three that we are going to focus on, and we're going to concentrate or focus on the impact, the relative impact that could have. Now, there's a lot of possible different scenarios here. It's not black and white here. There's a range. There's a lot of gray here. There's a lot of range of impact mutations can have, ranging from nothing all the way from no effect, okay, to small, all the way to complete non-function. And even on that scale, I need to expand that scale slightly, because just like you can have no effect and you can have non-function, there's also a little bit of opportunity for some improved or altered function. Okay, so we're on that scale here, and we're going to discuss what are the different scenarios that might give us some of these end results. Okay, I keep having to move this, so I keep waffling basically. All right, let's go. So we discussed a substitution mutation. Now let's consider substitution. I'm not going to draw out the whole genetic code or an example this time, or shall I? Actually, could I just copy what I did last time? Or could I just drag it down? Yeah. Nice. All right, so this is basically what we discussed last time, right? That we could have a substitution. Get rid of that. Okay, so our original sequence is in black, okay, and then our original mRNA sequence is in blue, and our eventual amino acid sequence, our primary structure, is in green, okay? And what we did was we changed, and all the changes are in red. So we swapped out the T for the C, and what we got was a potential 1 amino acid difference. So out of all the amino acids in this sequence, okay, remember, it goes on and on. This sequence could carry on and on for many amino acids in that direction until we get to the end of the polypeptide. And we changed 1 amino acid. Okay, but when we're considering the substitution mutation, what's the range of the impact it could have? Well, did we discuss this last time? I can't really remember. But because of the genetic code is degenerate, okay, or is there another word for this? Or redundancy. Because there's redundancy. So because of the degenerate or redundant nature of the genetic code, it so happens that more than one codon can code for the same amino acid. Okay, that's what that means. Right, so if we look at the genetic code, right, if we just zoom in there a little, okay, if we just zoom in there, we can see that there are a number of codons that could give us the same amino acid. Now usually if you change the last letter of the 3, you could get the same amino acid. Let's just see if that works out. So if I've got AT, right, so let's go A, so A, well, no, so if what do I end up with? UAC, right? Okay, let's try that again. If we go U, so we're thinking about the blue here, so let me just change my pen. Okay, we're thinking about the blue, we're following the blue codon, all right, so U, A, and C. U, A, and C gives us tyrosine. That's what we had in our original. But as you can see, if we go U, A, U, right, so if we had UAU, so if we had U here, that would mean we had an A here. Okay, so if the mutation was not not the second T to the C, not that, so let's just remove that. So if the mutation was different and we changed the last G, great, we changed the last G to an A, okay, so if we got rid of that G and changed it to an A, what would happen? That C in the mRNA would become a U, okay, U complementary to A, and UAU, if we follow that, U, A, U, we still get tyrosine. We still get tyrosine, even though we've had a mutation in the DNA. Even then, after that, even after that mutation, we have still got tyrosine as our first amino acid. Yeah? So, actually, we can just get rid of this. Okay, so we still get tyrosine, and this is because of the degenerate or redundant nature of the genetic code. Okay, so the least impact thing of a substitution mutation is no change, no effect on the polypeptide. That could happen, okay? But as we discussed last time, because of a substitution mutation, we could get one amino acid swapping out, okay? So we could get a change in one amino acid. So this is the other thing, okay? We could get a change in one amino acid, we get an amino acid swap for another one. Now, look at the other thing that might happen because of the substitution mutation. I'm kind of glad, just by chance really, that I ended up with this codon. So look at the other thing that could happen. Let's go back to our first codon, or triplet ATG. Okay, let's make a different change. Let's make a different change. That. Now this time, let's make a different change. This time, we're gonna have a UAG. I'm gonna change this G. Because of my mutation, because of the mutation that happened, that G is gonna change to a C. A, T, C. Let's look at the impact of that. So we get U. So in the mRNA, when that gets transcribed, we get U, A, and G. Okay, so now let's go back to our genetic code. Let's just clean it up a little bit. And let's follow that then. U, A, G. UAG is a stop codon. UAG is a stop codon. That's a termination signal to the ribosome. It tells the ribosome, stop here, this is the last amino acid, don't worry about it. You're done. Take a break. Okay, so, and therefore, at the point of this substitution, what would happen is no further amino acids would get added to the chain. So a substitution mutation could introduce a stop codon, which means most or any amino acids that would have been added after that might not have been added. So a premature stop codon is disastrous. Okay, that means so if you just got half of a protein, that means it's not going to work. Right? Randomly remove half of an engine. Randomly remove half of the components of your phone. Yeah? It might not work as well. Okay? So that's what a premature introduction of a stop codon does. Okay? So even something as seemingly insignificant as swapping out one base for another can have a quite a big range of effects. We can go all the way from no change to a change, just swapping out one amino acid for another. And guys, even if you change amino acids, if the new amino acid that you add has similar properties in terms of its R group, it might not cause any difference. Okay? But if you change the property of an amino acid, like from a negatively charged side chain to an amino acid with a positively charged side chain, that's going to have a bigger impact. So this is all the gray area that I was talking about. It's not gray because it's unknown, it's just, you just have to consider it at a number of different levels. Okay, so we've got change in one amino acid that could have a number of different things. I'll put that on there at some point too, because I like the discussion that we're having here. I think we're getting into it, so I don't want to lose that. So we could go from no change, nothing. Okay, we could change one amino acid, but we also could introduce a stop codon. Okay, some of the protein missing. Okay, that is going to be full-on non-function. Okay, it's definitely not going to work if it's got part of it missing. Okay, non-function. So as you can see, we've gone from no change to non-function. All right, let's just consider this further a little bit. The change in one amino acid. If we change one amino acid, if the new amino acid has the same properties, we might get away with it. Okay, little change. Okay, so it might affect the function a little bit, but it still might work. Okay, if the new amino acid has got the same kind of side chain, we might get away with it. Okay, however, the new amino acid might have very different properties, right? It could be hydrophilic, it could be, whereas the original amino acid had a polar side chain, the new amino acid has a hydrophobic side chain. Well, that's going to significantly affect the protein shape, right? Because that part of the polypeptide then is getting pulled to the center of the protein, right? Because of the non-polar interaction. So different properties, we have to start to appreciate that that's going to have more impact. Okay, and any given random change is going to be negative to the protein. Okay, all right, but there's a possibility for it to be some improvement as well, but that is very small. One last thing that we need to consider. Let's say that we do have this different property. Okay, let's say that we do have this different property. Okay, but let's think about this. What about location? What about location? If that amino acid is in the active site, it's going to have a big impact. Okay, so the final thing we have to consider is location. If the amino acid that's swapped out and changed is in a very important location for the enzyme function, it's going to have a big impact. But if the location, so the place that the amino acid is that's been changed, if that is in a kind of non-influential part of the protein, like the easiest thing is to think of an enzyme. So if it's in the active site, right, we're going to be in trouble then if we change an amino acid there. But if it's away from the active site, somewhere far away, that's kind of inconsequential, and that's a relative term obviously, but if it's not really involved in the reaction that the enzyme is catalyzing, again we might get away with it, so the impact might be small. So we have to consider also where exactly in the protein is this change going to happen. Alright, so I hope we have seen that even something as insignificant as a substitution mutation can have big impacts, can have no impact, and everything in the middle. Okay? And I think, alright, I guess we can continue a little bit. There's the last thing I want to talk about, which is the effect of insertions and deletions. Not together, obviously. Okay, insertions and deletions. Because while we're on this topic, we might as well think about what impact the insertions have. So when you insert one base, okay, when you insert one base, and whatever we say for insertions is going to be true for deletions too. Okay, let's just get our code again. Oops. T. I'll just tidy up a few things here. Okay, if we add one base to this, let's see what would happen. Okay, so I am going to add, let's just say I'm just going to add randomly here, I'm going to add an A. A has been added. Okay, now if I add an A, so what? What's the big deal? Well, the deal is quite big. Where's my red pen gone? Red color. All right. So what would happen? Well, if I add an A here, the first codon is, the first triplet is okay, it's still ATG. But the next triplet, instead of reading TTA, the next codon reads A, T, T. Oh, okay, what's the big deal there? Well, so ATT is, that's a change. Okay. So then the next set of three, instead of being AAC, the next set of three that are read by the ribosome are going to be A. So we've made another change. Okay, but then instead of the next set of three after that being GTA, it's CGT. So that's a change. And this will continue all the way to the end of the polypeptide. Okay, so I hope that we've seen that by adding a base, we have caused a change to the reading frame. That's what we call, we have added a change to, or we've made a change to the reading frame. This affects, this always, and there's no two ways about this, this always affects every single triplet / codon from the point of the insertion. And guys, the same thing would be true if we had deleted a base. So I'm just going to add that in there. Okay, so it affects every single triplet codon from the point of the insertion. Deletion, okay, changes, and by that I mean it changes, literally changes how, you know, the sequence of the triplets, the sequence of the codons. Okay, so we're not just talking about one amino acid being different anymore. We're talking from the point of that mutation, every single amino acid after that is different. Okay, so that, there's no two ways to look at that. That is always big impact. Okay, and there's no way that you make a, you know, a protein that is very, very different without causing a negative effect. Okay, big impact, always negative. Right? It's like getting some Shakespeare, right? You get something written by a famous author, you know, work of art, famous through the ages, and you randomly change 60% of the words around and expect any kind of improvement. That's not going to happen. Okay, so that's what we're talking about there. Right, next, what could happen? Well, yes, we could change. So the first thing is, that's a big impact. We could change much of the polypeptide sequence. And we were talking about one amino acid different potentially being detrimental to function. Imagine what you would do changing multiple amino acids in a polypeptide. Okay, so that's a big change. But also, it can get even worse. Not only could you change and swap out amino acids, multiple amino acids for different ones, not only that, you could actually introduce one of the codons that one of your new codons, the introduction of stop codon. That could also happen. So by changing all these triplets into other ones, it could be that one of the changes that are caused by that insertion or deletion, it could be converting a codon for an amino acid into a stop codon. So one of these new codons that comes into play because of the new reading frame could be a stop codon, prematurely terminating that polypeptide. Okay, so frame shifts are always big impact. Okay, I'll give you 10 seconds to think of a scenario where a frame shift doesn't cause a big change. Yeah, no, okay, well, I don't know, I can't think of one either. I mean, I was thinking that, you know, potentially if the frame shift happened at the very end of the protein, you know, most of the working bits would still be there, but then the stop codon would not be there, and you'd get this extra chunk of polypeptide added on to a protein. And yeah, I don't like the sound of that. So there we have insertions and deletions, and I think that's where I want to leave our discussion for today. So just chatting, you know, just hanging out, chatting about the impact of different types of mutation. I've got a niggling feeling that I've forgotten something important here, but yeah. All right, we'll leave it here, guys. That was fun. That was year 2, just a little bit of year 2 bio. I hope you've enjoyed chilling out and listening to that. Yeah, guys, keep on going. It's tough times, one foot in front of the other. And, you know, message me if you got suggestions for topics, videos, if you got questions. Yeah, keep on going, guys.